Alright guys, welcome to the final screencast of our cellular respiration series. Let's get right into the electron transport chain. So, I don't want you to concern yourself with too much of the biochemistry of this. People are getting PhDs based off the information you're looking at right now, so I certainly don't expect you to understand this completely, but I would like you to understand the overall concepts, which the electron transport chain is a series of protein complexes located in the inner membrane of the mitochondria, if we have multiple. After glycolysis and the Krebs cycles, we had 10 NADH and 2 FADH. For each NADH, in the electron transport chain, we get 3 ATP, and for each FADH, we get 2 ATP. Now, as you can see, we've got this diagram over here. We've got complex 1, 2, 3, and 4, and what's happening is that the electron transport chain takes place in the inner membrane space of the mitochondria. So remember that the mitochondria has two membranes. It has its outer membrane and its inner membrane. And the intermembrane space between the outer and the inner is where, so the inner membrane pumps NAD, the inner membrane proteins oxidize NADH and FADH, and they pump protons of hydrogen ions, into the intermembrane space. ATP synthase uses free electrons, the protons from the oxidation of NADH back to NAD and FADH2 back to FAD. And this series of protein complexes perform the oxidation of NADH and FADH2. It generates free electrons, which is used to reduce oxygen into water and which releases energy. This free energy, the electrons, are used to pump protons, the hydrogen ions, from the matrix across the inner membrane into the inner membrane space. This creates a gradient, which we've got a high concentration of ions on one side of the membrane and a low concentration on the other. Gradients, which we'll talk a little bit more about in osmosis, want to go towards being balanced. So the hydrogen ions want to return to the matrix. And ATP synthase uses this gradient built by the electron transport system to allow the hydrogen ions back into the matrix, and it uses the energy of this transport back across the membrane to convert ADP and a free phosphate into ATP. Again, this is a lot of information, and ATP synthase, this is only theoretical how we think ATP synthase works. There are people in labs somewhere right now trying to figure out exactly how ATP synthase works. But we do know that ATP synthase allows the protons back across the membrane, converts ADP into ATP. So, all in summary, we had our 10 NADHs from the Krebs cycle and glycolysis and the transition phase between glycolysis and the Krebs cycle. We get 3 ATP for those 10 NADH. We've got 30 ATP in the electron transport chain. Then the 2 FADHs, we get 2 ATP for those, and that gives us 4 ATP. We have our 2 ATP from glycolysis and our 2 ATP from the Krebs cycle, which would get a total of 38 ATP total from one invested glucose molecule. And we have a lot more than just one glucose molecule in our energy stores. So this is a very efficient reaction when it is under oxygen. But there is another path for glucose to be utilized into energy. Fermentation. We have two types of fermentation. We have lactic acid fermentation and alcoholic fermentation. This is an anaerobic process. It does not use oxygen. So our electron transport chain, our Krebs cycle, all require the presence of oxygen, and it is a highly efficient process. Fermentation is a less efficient process, and it is with that when the cell lacks oxygen. So lactic acid fermentation. It occurs in humans. No gas is produced. There is no gas byproduct. This is unusual. It's the only cellular respiration process that doesn't produce gas as a byproduct. No oxygen is required for it. It uses the pyruvate from glycolysis. It converts the pyruvate into lactate, and it oxidizes NADH to NAD. NAD is the necessary molecule for glycolysis, which, as we know, is the first step in our cellular respiration pathway. So it replenishes the NAD for glycolysis to take place. But we don't get any energy production out of it. Then there's alcoholic fermentation. Alcoholic fermentation, pyruvate is converted into a chemical known as acetaldehyde. NADH is used to convert acetaldehyde into ethanol and alcohol. NAD is replenished for glycolysis again in alcoholic fermentation. Alcoholic fermentation takes place in organisms which are facultative anaerobes. A good example is our old friends yeast. Alcoholic fermentation takes place in yeast. So to summarize fermentation, if oxygen is present, the cell will bypass fermentation. If there is a buildup of pyruvate, fermentation may occur. This can occur during intense exercise. We lack what we need to continue aerobic respiration, and anaerobic respiration will occur, and this is why muscle fatigue sets in. If oxygen is not present, the cell will replenish NAD to glycolysis using fermentation. So if there's no oxygen ever present, then fermentation is the pathway which we will use pyruvate in, or pyruvic acid. Now, to help some of you with your test tube lab design, I know some of you had trouble starting on this. Here is an ideal way to test our two hypotheses. So one of our hypotheses is testing sugar content as a variable, and so we will add 25 grams of yeast to each test tube. Then we set up each test tube with 20 mLs of water and the varying grams of sucrose. So we are varying the amount of sucrose in each solution. We're keeping our yeast constant, and we have a control in test tube 1. A control is to ensure that there's no other variables being tested in the experiment. So if we just mix yeast and water, there should be no CO2 production. Once you've put 25 grams of yeast in each test tube, or 0.25 grams of yeast in each test tube, put a stopper in the test tube, gently mix the solution by inverting it a few times, turning the test tube upside down and back up. Leave the stoppers loosely on the test tubes, and then time the reaction. Record the time you observe gas being produced in each test tube. At 15 minutes, measure the bubbles using a ruler or froth at the top of the test tube in centimeters and record that data. Then, to test our second variable, and this should be We'll correct this right now. So we're testing yeast content as a variable. We put 20 mLs of a 20% sucrose solution, warm sucrose solution, in each test tube. Then we're going to, we need to adjust that yeast value. So we need to have 0.0 grams, 0.1, 0.15, 0.3, 0.4, and 0.5. Alright, so now we have varying amounts of yeast. Again, once you've added the yeast to your test tube, put a stopper, gently mix the solution, leave the stoppers loose on the test tubes, and be careful with test tube number 12. The stopper might pop off of the test tube. Time the reaction. Record the time you observe gas being produced in each test tube at 15 minutes. Measure the bubbles or froth at the top of the test tube in centimeters and record the data. That's our screencast for the night. I hope you have a good evening and good night.